Hello everyone. Today I'll be talking about how we can use clustering methods to identify patient subtypes in neuroimaging data. This is important because by finding patient subtypes, we can better understand the disease that we're looking into and its effects on patients. Specifically, we'll look at how clustering helps identify different patient profiles based on structural neuroimaging, and at the end I will have a tutorial that you can use. So, why do we want to look into patient subtypes? Well, this is due to disease heterogeneity. This means that the same condition can display different traits, and this can be seen as symptoms. And this disease heterogeneity can be affected by different things such as genetic traits, environmental factors, or immune response. For example, in my study with long COVID patients, even though my patients and my controls showed no statistical differences in the initial analysis, the patients reported cognitive symptoms that impacted daily life, which raised the question of whether there are subtypes to uncover from our data. And to find these subtypes, we use clustering, which is a process of grouping a set of data objects, which in our case are patients, into multiple groups or clusters, so that each object within a group has a high similarity, but they are very dissimilar to objects in other clusters. It is also worth mentioning that clustering is an unsupervised learning method, meaning that we do not know the groups in advance. We have no previous knowledge. A powerful method used for neuroimaging is hierarchical clustering, which builds a hierarchy or a tree of clusters that it's called a dendrogram, and we can see one currently on the screen. And there are two main types: agglomerative or bottom up, which starts by treating each data point as its own cluster and merges them step by step. But then we also have divisive, which is a top down, which it starts with the one larger cluster and splits it iteratively until we have single objects. This tree-like structure helps us visualize the relationship between the clusters, and whether using an agglomerative method or a divisive method, it is necessary to measure the distance between two clusters. And there are several ways to measure the distance, which are called linkage methods. Each method would have its own way of defining how two clusters are similar or different, and these choices can impact the final clustering result. We're now going to talk about these linkage methods, and we'll start by single linkage. Single linkage finds the closest points between two clusters and uses that as the distance between the clusters. For this reason, it is sometimes called a nearest neighbor clustering algorithm. And this method can result in a chain-like cluster where even distant points from the same cluster may be connected, making it sensitive to outliers. Here we have the function for minimum distance, and here P is a point in a cluster I, and P apostrophe is a point in a cluster J. And then we have P minus P apostrophe, which will represent the distance between these 2 points. Then we have complete linkage, which measures the distance between the furthest points in each cluster, which can help avoid chain-like clusters from the single linkage. It is also called farthest neighbor clustering algorithm. And this method is more sensitive to outliers, as the maximum distance can be greatly influenced by extreme values. This formula is very similar to the single linkage, but we take the max instead of the minimum distance. Next, we have the centroid linkage that calculates the distance between the centers, or we can also call them the centroids, of two clusters. It is less sensitive to outliers than complete linkage because it is based on the average location of the cluster, so the middle point. But may not always work well in non-spherical clusters for the same reason. Here we have the function for mean distance, where mi and mj are the centroids or the mean position vectors of clusters ci and cj, respectively, and the formula will calculate the Euclidean distance between these two centroid points. Next, we have the average linkage, which measures the average distance between every pair of points in the two clusters. This method is a balance between a single and a complete linkage by reducing the sensitivity to outliers, while still taking the overall structure of the clusters into account. Here we have the function of the average distance, and where ni and nj are the number of points in cluster ci and cj respectively, and the sum is over all the pairs of points where p in ci and p apostrophe in cj. And then finally, we have Ward's method, or Ward's linkage. This is a method that aims to minimize the variance within clusters, and it's often the default method in clustering because it balances clusters' compactness and separation. Instead of directly measuring the distance itself, it merges clusters that result in the smallest increase in the error of some squares. And here we have a function for the distance, Ward distance, where ni and nj are the number of points in clusters ci and cj respectively, and mi and mj are the centroids of the two clusters. This method computes the square Euclidean distance between the centroids, weighted by the cluster size, and this approach is particularly useful in neuroimaging, where we want well-defined and meaningful subtypes that minimizes internal variability. And at last, we would have a measure of cluster quality, and because once we're done the clustering, we need a way to evaluate how good our clustering results are. The silhouette coefficient is a popular metric as it gives us a way to assess how well an object is clustered. It is going to compare the average distance within the cluster, which in this case would be AP, with the average distance to the nearest other cluster, which would be BP. It ranges from -1 to 1, where higher values indicate a better clustering. If the value is close to 1, the point is well clustered, and as the score close to 0 suggests that the point is a near boundary of two clusters, and a negative score suggests it's in the wrong cluster. So now that we have all of this information, let's apply these clustering methods to real neuroimaging datasets. So for this, I have the following tutorial, which demonstrates how to use hierarchical clustering on neuroimaging data to explore patient subtypes. And for this, we're going to process gray and white matter data, applying the clustering technique, and then we're going to visualize our results. So first of all, we need to import all the necessary Python libraries and for processing neuroimaging and for performing the clustering. These ones are going to include the NumPy, Pandas, Nibabel, Matplotlib, PyPlot, which is going to help us visualize, and also Nilearn. For this, after we have our libraries, we need to set up our directories. So these ones are gonna be the ones that store the MRI data, the atlas files, and also where we want our results to go into. It is important to know that the data from this tutorial are derivatives from the FSL, BBM, and TVSS analysis that I did for my long COVID project. So first, we're going to start by preprocessing the data, which in this case, gray and white matter are preprocessed in a similar manner. And on this step, the MRI data, either white matter or gray matter, are parcellated using an atlas. And I would specifically explain the gray matter, how we do it, and then the steps repeat for white matter. So first, for the pre-processing, we would need to load our gray matter data as well as our atlas, which in this case, we're going to use the AAL atlas, which contains 120 regions and also the labels for the regions of this atlas. So here I have a visualization. We visualized our brains. This is our MRI data right here. So we can see this is our brain data. And then we can also visualize our atlas. This is a good practice to look into your data. Once we visualize our data, we extract the gray matter data the underlying gray matter data that we have, we extracted with using the function getFdata. And this allows us to manipulate the data as if it was a NumPy array. And after that, we iterate through each subject, which represents our fourth dimension in our data, and we process each region in the atlas. So for every region, we mask it and extract the voxel values. We then calculate the mean signal of all the voxels within that region, which becomes the region signal value. It is crucial to exclude the zero values from our data because they represent non-brain areas such as empty space, and they are not actual brain tissue. So if we include these zero values, it could distort the signal, leading to inaccurate results by artificially lowering the mean signal. And after we calculate this, we organize the data into a data frame, and we add the subject IDs as a distinct column, and then we visualize it here, and we can see that our data frame looks like this. We have our column names, which are each region, and we have the mean signal value for each region for each patient. In this case, here it has our control data as well as our subject data, which is an S. And for further processing, for further analysis we wanted to do, I separated in controls and subjects, and then I saved it in an Excel file. This will allow us to retain the data for further use, as I said, and it can also be easy to share if we need to. And then for the white matter pre-processing, we do the same steps. The only difference is that we use a different atlas, and this one is dedicated for white matter tracks. And I have here the visualization. In this first one is our patient's data. We can see it right here. And then we have our atlas data, where we can see it is divided by different regions, and this is what we use to parcellate it. We follow the same steps that we did in gray matter, and we end up with our atlas, with our data frame. We end up with our data frame, and we can use it for the hierarchical clustering. Now, after we pre-process the gray matter and the white matter, we merge them for the clustering analysis, and then we drop the IDs before clustering. For our model, we're going to use the linkage method of the word linkage method, and we then visualize it in a dendrogram. As you can see, we only see a dendrogram. It doesn't have any division in colors or anything like that, but we can do it by determining the optimal partition distance threshold. And for that, we're going to use the silhouette score, which we learn is a evaluation metric for clusters. And what we do here is that for each distance, that value that we extracted from the hierarchical model, we calculate its corresponding silhouette score. And we then select the best distance threshold that yields the highest silhouette score. as it indicates the best partitioning. And here I have a graph to demonstrate that. So we would have each distance threshold that we acquired from the model, and then we will calculate the silhouette score for each one of them, and it would give us which one is the best threshold. It is important to mention that here our silhouette score is 0.1 or 0.10, which, if you remember, means that our clusters might not be greatly separated, and that can be for many reasons. But if we recall, closer to 0 means that some data objects or some patients can go are in the boundary in between the two clusters. And we can see that by applying a principal component. We extract our subjects and our clusters and our subjects, who is in each cluster. And first of all, we can plot it, and we can see the threshold that we use by using the color threshold, and we put the one that was the best. And then we can see the separation in color of the two clusters. Once we have that done, as I said, we can visualize it using principal component analysis to see where our clusters lie in 2 dimensions. And as we recall, our silhouette score was 0.1, which means that some of our clusters or some of our data objects or patients in this case are in the boundary between the two clusters, which can be represented here. And now, once the clusters have been assigned and we have looked into our data, we can use this cluster dictionary that we created over on here, and we can use it to do any further analysis to compare patient subgroups.